Hello everyone, I would like to thank the International Continence Society for giving me the opportunity to present my work today.
We have nothing to disclose. So many organs in the body have either left or right dominance in their function, and the most popular example we see is people with left hand dominance or right hand dominance. The regulation of the physiological functions of the urinary bladder requires coordination between a neuronal circuit within the brain and the spinal cord. Our group have previously noted a left laterality in canine bladder in response to pelvic nerve stimulation. And so this study is a part of a larger project that aims to re-innervate the urinary bladder after long-term decentralization by performing nerve transfer surgeries in a canine model. So a question that we could ask here is, if we had to choose a side to re-innervate the bladder for a return of function, is there a better side? So since the bladder receives innervation from the right and the left sides through the spinal cord and the pelvic plexus, the goal of the study aims to examine if stimulations of either the spinal cord or the pelvic plexus causes a greater bladder contraction on the left side versus the right side. And to test for this, we have performed functional electrical stimulation of bilateral lumbar L6 through S-sacral S3 spinal cord roots and the left and right pelvic plexuses. Following that, the maximum detrusor pressure changes were obtained for left and right side for each spinal root and for the pelvic plexus. Maximal contractions produced from electrical stimulation of L6 through S3 spinal roots were recorded in 44 animals. Left root and right root evoked contractions are displayed on the y-axis and the x-axis respectively. Each point represents the maximal contraction obtained from stimulation of either the left or right spinal root in a single animal, whichever produced the strongest bladder contraction. The dominant side for each spinal root and pelvic plexus in each animal was determined by calculating the percent difference, which was 25% between the average of the left and right stimulations. Bladders were considered left or right-sided if differences are greater than or less than 25% respectively. If the differences were within 25%, bladders were considered bilaterally innervated. Differences between left and right maximum detrusor pressure was evaluated at each individual spinal root L6 through S3, and upon the L6 spinal root stimulation as shown in figure A, of 12 animals tested, 5 of the bladders were left dominant, 5 right dominant, and 2 were bilaterally innervated. In figure B, upon the L7 root stimulation of 28 animals tested, 10 had left-sided bladders, 10 had right-sided bladders, and 8 were bilaterally innervated. Stimulation of S1 spinal root in 36 animals showed that 16 animals were left dominant, 14 were right dominant, and 6 were bilaterally innervated as we see in figure C. Similarly, S2 root stimulation indicated that out of 32 animals tested, 12 were left side dominant, 10 were right side dominant, and 10 were bilaterally innervated, as shown in D. And finally, upon the S3 root stimulation of 18 animals tested, 8 had left dominant bladders, 7 had right dominant bladders, and 3 had bilaterally innervated bladders as shown in figure E. Here, the maximum detrusor pressure of any spinal root tested, L6 through S3 left and right from 44 animals were recorded. Similarly to the previous graph, percent difference between left and right maximum detrusor pressure for each animal was calculated. And from the 44 animals we tested, about 38.6% of the bladders were left dominant, as we see here. 27.2% of the bladders were right dominant, and the remaining 34% were bilaterally innervated. Maximal contractions produced from electrical stimulation of the pelvic plexus were also recorded in 19 animals. Left and right plexus evoked contractions are displayed on the y-axis and the x-axis respectively. And each point represents the maximal contraction obtained from the stimulation of either left or right pelvic plexus in a single animal. The percent difference between the average of the left and right maximum detrusor pressure for each animal was also calculated, and from the 19 animals we tested, about 42.1% of the bladders were left. Overall, this study and the data provided us with the evidence for the existence of asymmetry of the bladder function in the normal female dogs. And as indicated in the table, about 1/3 of the animals we tested showed left dominance in their bladders, 1/3 right dominance in their bladders, and the remaining 1/3 were bilaterally innervated, as we see here in the table. And although the current observations were obtained from normal animals, the determinant left and right dominance of the bladder function might potentially provide us with the basis for understanding the consequences of lateralization of bladder innervation in patients with bladder dysfunction. At the end, I would like to give special thanks to my mentors, Dr. Mike Ruggieri and Dr. Mary Barbe, to all my colleagues at the Ruggieri and Barbe labs, and to our collaborators, and for the funding source.